We're going to talk about the end of chapter 28, and then we're going to move into development in chapter 29. So chapter 28 is looking at the hormones involved in the menstrual or uterine and the ovarian cycles. This is on page 5 in your chapter 28 outline. So take a look at that, and you'll see on there there are the menstrual cycle or uterine cycle, and the ovarian cycle needs a definition. And so the definitions for those, the uterine cycle is the 28-day cycle of the uterus when it goes from shedding the functional zone of the endometrium all the way through the rebuilding and glandularization, vascularization of that functional zone, and it's ready now to hold a blastocyst. The ovarian cycle is a monthly series of events that's associated with the maturation of the egg, or the oocyte. And so we saw it going from the primordial egg all the way through to the tertiary follicle that ruptures during ovulation and releases that oocyte hopefully into the uterine tube. There are 5 hormones that I want you to be aware of. The first that you see at the top of this chart is the gonadotropin releasing hormone, and that's the one you see here coming from the hypothalamus. And when the gonadotropin releasing hormone is released, it's going to stimulate the anterior pituitary gland to secrete FSH and LH. It starts this process at about day 1 of that cycle. FSH actually does what it says it does in the female side, so it's follicle stimulating hormone, and you'll notice that it stimulates the developing follicles to move from that primordial follicle that we talked about all the way through those stages: primary follicle, secondary follicle, tertiary follicle. It also will stimulate the ovaries to produce estrogen. LH is going to stimulate the secretion of progesterone by the corpus luteum. So those are the important things. Estrogen and progesterone. Estrogen, as its levels go up, you'll notice that what it'll do is it'll actually increase the frequency of the gonadotropin releasing hormone, which will stimulate a huge surge in FSH and LH, and that's where we got the ovulation to take place. If you remember, when we looked at that chart, we saw a big peak in FSH and LH right before ovulation, and estrogen helps to play a part in that. You'll also notice that estrogen is involved with a significant number of things. It affects the central nervous system, it stimulates bone and muscle growth, it's responsible for the female secondary sex characteristics, it's going to maintain accessory glands and organs, such as the mammary glands. And it will help with the stimulation of endometrial growth and secretion. It helps to maintain that uterus. The progesterone that's being released, that is its primary function, is to stimulate endometrial growth, and it allows for that uterus to remain in place throughout the pregnancy. The mammary gland is a specialized sweat gland, so it actually started out as a sweat gland and then had some modifications. There are a couple of pieces that you should know. It is, if you look at it, it looks like a whole cluster of grapes. That's often referred to as the lobe. And then each individual grape that you see on there is a lobule. The lobules are all connected by a lactiferous duct, and the lactiferous ducts join together and become the lactiferous sinus. This breast tissue is found in front of the pectoralis muscles, the pectoralis major and the pectoralis minor. It is located just underneath the dermis, so it's actually in the hypodermis, and you can tell that because you have this lovely adipose tissue here. There's yellow bubbles that you see there. When a woman isn't pregnant, she has an inactive mammary gland, and that's the one that you see at the top of the right-hand side. There are a couple of hormones that help to convert the inactive mammary gland into the active mammary gland, and you can see that active mammary gland here. Prolactin is the primary one that is involved in that change, in that conversion from inactive to active mammary gland. The milk is actually produced within the lobules. There are secretory alveoli. They look similar to the alveoli in the lungs because they are essentially these small cells that are formed around these open spaces, and that's where the milk would end up. So the secretory alveoli are responsible for producing milk. So that ends the information on reproduction, on the female reproduction, and now we want to move over to development and inheritance. And on your chapter 29 outline, you should have that printed out by now, you will see that at the very top there is activity 1, which is doing some definitions. So go ahead and go to page 1077 and look up those 5 terms, see what you can come up with, and then pause the video. When you've got those definitions, then we'll come back and we'll start with letter B and look at conception. So our objectives to start out with is going to be to describe the process of fertilization, and then we're going to explain how we use cleavage to divide a zygote until it eventually becomes a blastocyst. We're going to talk about that process of cleavage. Just to show you some pictures, when we use the term embryo, when we use the term fetus, embryo is from that zygote all the way to the 8th week of conception, and you can see there is a lot of development that has happened in those first 8 weeks. And once we've gone past the 8th week, and we are now in the 9th week, from there on we will refer to that developing baby as the fetus. Okay, so embryo and fetus is what we will use there. Conception. This picture absolutely amazes me, particularly when you see the definite proportional differences between the sperm and the egg. So we have numerous sperm that are trying to crowd around and get into the egg. The process of conception is essentially the same thing as fertilization. This is when the nucleus of the sperm is fused with the nucleus of the egg. So let's take a look at how that process goes. The egg actually has a protective mechanism around it. It has 2 layers. The first is a jelly-like layer called the corona radiata, and on here it's this yellow layer that you see. And then it also has underneath it, it has a pink layer called the zona pellucida. So we'll just put an arrow there as well. It is tough for that sperm to be able to protect those 2 layers. We want to make sure that we only have one sperm penetrate that. But let's take a look. We have some terms here that we need to talk about. The first is capacitation. Capacitation is the final maturation of sperm. It allows the sperm to swim rapidly to the egg. Capacitation begins when the sperm mix with the seminal gland secretions in the ejaculatory duct, and then as the sperm pass through the urethra and in of the penis and into the vagina in the female, we end up having complete capacitation. So at this point, the sperm is really ready to go. Remember, at the head, at the very tip of the head of the sperm, we have the acrosome. And that acrosome is filled with hydrolytic enzymes, and those enzymes are actually going to break down the barriers that are around the egg. So we have acrosomal enzymes being released here on the illustration, and they will start to break down the corona radiata, and eventually they will break down and be allowed into the zona pellucida as well. This is not a single sperm activity, but actually would require multiple sperm to break down the corona radiata. So if you only have one sperm that reaches the egg, it's not going to get fertilized. Eventually, a single sperm manages to penetrate through that zona pellucida, and as soon as it does, it changes this outside layer so that the hydrolytic enzymes no longer function. So it allows one sperm in, and that's it. When the egg and the sperm nucleus fuse, this is when we have a completion of meiosis for the egg. So the egg doesn't actually complete meiosis until we have this fusion. And so monospermy means that we only have one sperm per egg that actually penetrates. And then when the egg and the sperm nuclei fuse, we end up with a zygote. And that is the first cell of life. This zygote is diploid because it has 46 chromosomes in it. 23 of those came from the sperm, 23 of those came from the egg, and that allows for a diploid cell for the first cell of life, just like all of the cells in your body except either your egg or your sperm. Before we go on, I'd like you to see if you can explain the difference between a monozygotic and dizygotic twins. So I'll give you a moment to think about it. Monozygotic twins start out with, hopefully you figured this one out, start out with one zygote. Mono means one. And somewhere early in the cleavage process, the cells split and we end up with two embryos. So embryo 1 and embryo 2. Because they came from the same zygote, they're going to have identical genetic material. This zygote was the original 46 chromosomes, and then this embryo and this embryo have those same 46 chromosomes, which will allow them to differentiate. So monozygotic twins, we also often refer to them as identical twins. Dizygotic, di means two, so we have mom released two eggs, and they both ended up being fertilized, so we have zygote A and zygote B, and from both of these developed an embryo. Now, in this case, they have their own unique genetic material because zygote A came from one egg and one sperm, and zygote B came from another egg and another sperm. So these we often refer to as fraternal twins. Okay. Cleavage. In the first trimester, we have a huge amount of development that occurs, and cleavage is the set of cell divisions that happens from day 0 all the way through until we end up with a blastocyst. And so you can see that we start out at day 0 with a single cell, the zygote, and within the first 12 hours that cell already divides and becomes a 2-cell stage, and within a day or two we're at the 4-cell stage, and then very quickly those divide again, 8 cells, and eventually we end up with something called a morolla. And a morolla is when we have so many cells that have divided that we've kind of lost track. It's not easy to count them. And I believe morolla comes from the Latin or Spanish term that sort of is referring to a raspberry or a blackberry. And if you think about it, it kind of looks like that. It has all of these little cell bubbles that are there. Each cell is diploid, so each cell has undergone the process of mitosis to divide. So there are still 46 chromosomes in each cell. But now these cells, you'll notice, if we look here, the cell in the 2-cell stage compared to the cell in this morolla, the 2-cell stage are significantly larger. Why are they bigger? Because remember that egg was huge. Back on that picture that we showed you a couple slides ago, had that huge egg, lots of cytoplasm. Because this cleavage process is happening so quickly, in 4 days we went from a single cell to 16 or 32 or even 64 cells. It's so big so quickly that there isn't time for cytoplasm generation. And so the nucleus divides and the cytoplasm is divided into smaller and smaller packets. So every cell has 46 chromosomes in it, but its size is being reduced because it hasn't had a chance to grow. By day 6, day 7, we now have something called a blastocyst, and that's on your outline as well. The blastocyst is when we first begin to see some differentiation, and differentiation is when we have cells that look a little different are going to become something different. And so on this blastocyst we have the outer blue cells, which are called the trophoblasts, and they're going to become the extra embryonic membranes. They're going to become the support tissue for the developing embryo and fetus. So they'll become the umbilical cord, they'll become the placenta, they'll become the amniotic sac and the amniotic fluid. The pink cells that you see in there, that is called the inner cell mass, a really creative name. And that inner cell mass, so this little few cells that you see here, that's what eventually developed into you. Okay, this is what will become the embryo, become the fetus. So blastocyst is made up of 2 parts. It's approximately 100 cells by the time we get to this stage, and it has been moving. Let's take a look at the next slide. You can see here this comes right out of Martini. So the amazing thing on this one is you can see when fertilization occurred up in that upper third of the uterine tube. You can see that cleavage is occurring all the way down as we move down through that uterine tube. And by the time we get to the uterus, we are at that blastocyst stage, and it's ready to implant. Implantation is exactly what it says. It is implanting that blastocyst into the uterine wall. It's small enough still that it actually, the whole thing kind of sits in the uterine wall. Let's see if we have another, there we go, we have another picture of that actually happening. So we actually have that sitting in that wall for you. Okay, so that's the trophoblast that's happening there that occurs somewhere between week 7, week 10. The inner mass cells separate from the trophoblast at this point, and we start to have an amniotic chamber. It's actually filled with fluid. We're going to look at those extracellular membranes in just a moment. I do want to talk about one hormone that's being secreted at this point. The trophoblast is now starting to secrete something called human chorionic gonadotropin, and that is what the pregnancy tests use to determine if somebody is pregnant. So they're looking for human gonadotropin when you do a urine test or a blood test for pregnancy. It is only secreted by this trophoblast and eventually by the placenta. And so a non-pregnant woman obviously wouldn't be secreting that. The amazing thing is, as we have gotten more and more sophisticated, we are much more able to see smaller amounts of this hormone on these test kits. And so that's why women are now able to tell that they're pregnant sooner than what they did 25, 30 years ago. I know when my mom took her pregnancy test, it had to be, typically you were already pregnant for 3, 4, 5 weeks. Now it seems like they know it within that 7-day window. You can pee on a stick and find that information out. All right, so this is implantation. We're going to continue to move along, and the next objective is to identify future systems of each germ layer. We're actually going to wait for that for just a second. The first thing we're going to do is pull up the next objective, which is to recognize the importance of each extra embryonic membrane. So these extra embryonic membranes, and I know I skipped activity 2, we'll come back to that in just a minute. So if you flip to page 2 in your outline, I have these extra embryonic membranes listed. There are 4 of them: the yolk sac, the amnion, the allantois, and the chorion. Sorry about that. That's one of the fun things about taping this when you're at home. You hope nobody calls, but you never know. All right, so we have, as I said before, we have these 4 different embryonic sacs. The yolk sac is going to help to deal with a little bit of nutrition. So it provides food before the embryo has an established support system. So before the chorion really gets established, before the placenta gets formed, it still needs to be able to provide some nutrients for this growing embryo, and so the yolk sac does that. The yolk sac will also eventually become a part of the umbilical cord, and then eventually it disintegrates. It doesn't hang around for the entire 40 weeks of pregnancy. The amnion, so you see the amnion here, this becomes the sac around the fetus. The fetus is a little tough to see. It's the little black striped line right above the yolk sac. That is supposed to be your amnion. I'm sorry, that's supposed to be your embryo. And then the amnion is the blue fluid that's above that. So the amnion creates a sac around the growing fetus, and you'll see it a little better on the next slide. And it provides a buoyant, protective fluid layer all the way around this developing embryo. And this is when the woman actually delivers a baby, and we hear about her water breaking. The membranes of the amnion actually break, and that allows the amniotic fluid to be released, and typically that's going to bring the baby's head down into the birth canal. You do not see the atlantis in here, but it's actually going to start to develop into, we're beginning a umbilical cord. It's initially called the body stalk. So let me see if I can draw a line. So it's right about in there is where you would see this atlantis. Okay, so this is going to become part of the umbilical cord. And then the chorion, right now you see it all the way around this fetus, this purple out here. This is the chorion. This is, sorry, down in here. This is going to become the placenta. So here we can see at week 4 a little more development. Again, you can see the amnion has started to truly develop. We don't have a placenta yet. It's still just the chorion, but it's beginning to develop into that placenta. The yolk sac, you'll notice, is starting to diminish, and we're starting to see some differentiation. So you have this embryonic head fold. You're actually working on starting to develop the gut of the embryo, and so on. At week 10, now we have placentation, so we have a placenta. Okay, this placenta with a body stalk has now truly become an umbilical cord. Circulation. One of the things that we want you to be aware of when we take a look at this is that you can see it here, and then we'll show you on the next slide as well. The placenta was completely developed from the embryo. Okay, and it does not mix the blood between the fetus and mom. They are their own separate circulatory systems. The circulation is going to come close, so there's capillary beds in both, but it doesn't actually, blood doesn't cross over from one to the next. So let's take a look at that next picture. Here you can see again, where the placenta is, you can actually see the blood vessels, and there's this space around these villi, these chorionic villi of the placenta, where mom's blood will actually fill that empty space in there, and then diffusion can happen very easily. But it's not a mixing of blood. That is a test question. Do mom's and baby's blood mix at the placenta? And the answer is no. Another question that could be asked is, does mom produce the placenta, or does the embryo produce the placenta? And the answer is the embryo. The placenta is completely made by the embryo. Hormones of the placenta, we already talked about human chorionic gonadotropin. So I want you to make sure you're comfortable with that one. That's going to help to maintain the placenta, make sure that we don't lose the endometrial layer of the uterus. Relaxin is also produced by the placenta. That's going to help to relax the ligaments in mom's joints, which is going to make it easier when we get to delivery, because the pubic symphysis, which is the ligament that's between the two pubic bones, is going to be a little stretchier. It's going to help open up that birth canal just a little bit wider. It's also what gives mom her pregnant waddle, because her joints truly are a little looser than what they typically would be. The placental lactogen and the placental prolactin are going to help with mammary gland development and milk production. And then progesterone and estrogen, those are the sex hormones. Again, they're being produced by the placenta to maintain that uterine lining, because we know that when those hormones drop, when progesterone and estrogen drop in levels, that's when the uterine lining sheds, and we obviously don't want that to happen. So now we are at gastrulation. Go back a page in your outline. Okay, so go back to page 1, and you'll see activity 2. It asks you to use the information on page 1083 and 1084 to define gastrulation, and then identify the components of at least 4 organ systems and say which germ layer they form from. So, for example, in the endoderm, well, let me go back. In the mesoderm, one of the examples is the skeletal system. So the skeletal system develops from the mesoderm. So pause the video, take some time to answer that, and then when you're ready, come back and we'll talk about gastrulation. You're back. All right, so gastrulation is basically when we start to see some differentiation. These are when cells actually differentiate and they no longer look identical. We start to see, and the first layers that we see is an ectoderm, an endoderm, and a mesoderm. And so each of these is a layer. The ectoderm was going to be the outer layer, the mesoderm is going to be the middle layer, and the endoderm is going to be the internal layer. And so if you look at the chart on the next page, if you had some problems answering that question about what you can find in the ectoderm, mesoderm, and endoderm, hopefully from this one you can find that. You can find some different structures that would be found there. All right. Differentiation typically occurs by the end, starts to occur by the end of week 3. Obviously, that differentiation process continues throughout the development process, because throughout development, they're constantly producing new different types of cells. So different genes are being turned on in different cells to make them into new things. By the end of 8 weeks, all of the major organ systems have formed. They still need to mature and develop, but they're all there. Every single one that we call a major organ system that we study in A&P 1 and 2, are the beginning of development has started. Also, arms and leg buds have started as well. From the first trimester, when this embryo and fetus has done a whole lot of developing, we're next going to move into the second trimester. At this point, the embryo is definitely called a fetus. And this is where we see a major increase in size. So the first trimester is all about developing, getting everything there. Second trimester is about making everything bigger. By the end of the second semester, this fetus is going to be about 600 times larger than what it was when it started out. So it is a huge time of growth. You'll also, a pregnant woman will say that this is when they start to feel movement. So the fetus and uterus are growing large enough, and the fetus is active, and the movements inside there are starting to be able to be felt by the mother. Third trimester, this is when we have the largest weight gain. That is for the fetus, not for mom. The fetus starts out at about 1.4 pounds and should end up somewhere between the 7 and 8 pounds at the end of the third trimester. Now, obviously, those are your normal parameters, and you have some that are going to be smaller than that and some that are going to be significantly larger than that. To me, the most interesting thing about watching this pregnancy is looking at what happens to mom's other systems. So let's take a look at this picture. And on the left, you'll see a female mid-sagittal cut, so you can look in there and see where all of the abdominal organs and pelvic organs should be. And on the right-hand side, you'll see now what's happening since this full grown, this is at full term, so you'll see where this fetus is squishing all of mom's organs. So you'll see all the intestines that were here are now all crunched up in there. So it does explain why it's a little more difficult for mom to eat a whole meal. It also will explain why breathing is a little bit more difficult, because here you can see where that diaphragm is, and the non-pregnant side, and the diaphragm is completely lost over here on the pregnant side. It's been pushed up farther into that thoracic cavity. So when we look at these different respiratory system, mom's respiratory rate definitely increased because she is getting oxygen in for both herself and for the fetus, and she has to expel waste products and carbon dioxide for both herself and the fetus. So we see an increase in respiratory rate. Depending on how much that baby is pushing up on the diaphragm, we may also see an increase in tidal volume so that mom can take a bigger breath more often. That will just simply increase her gas exchange activity. Cardiac, there is an increase in blood volume, about 50% of the blood volume that you would normally see, 50% more than what you would normally see, because it's not only supplying all of mom's organs, but it's going to this placenta, and we have lots and lots of blood that's there to exchange materials with the fetus. Kidneys, because of that increase in blood volume, we're also going to see an increase in urine output. It's problematic for mom. So we have more blood volume, which means the kidneys are going to be doing more screening, but if you look at the bladder down here, her bladder's being compromised. It's significantly smaller. Again, if we do a comparison here, take a look at what's going on there. So that blood volume's gone up, kidneys are screening more out, producing more urine, but the capacity of the bladder is smaller, and it's got this constant weight pushing down on it. And so that's why mom ends up feeling the need to urinate more often, even though the volume that she urinates may be smaller than normal. Finally, joints, again, the ovaries and the placenta are both releasing relaxin. That's going to make the ligaments loose. It's really great at the pelvic cavity, but it can make mom feel clumsy, and again, give her that kind of pregnant waddle. Labor and delivery, I'm not going to go into the specifics of this one. If you'd like to take a look at that, if you're not aware of this, I know some of you are in the OBGYN portion of your nursing rotations, and some of you have had children or have spouses who have had children. So if you're not familiar, feel free to take a look at it. There won't be a lot of questions on this particular illustration on the exam. However, at the very end of the lecture outline, there are questions that deal with this chart. And so this you should feel very comfortable with. So you'll see on page 3 of your outline, there are 5 questions, starting with how does the growth of the fetus contribute to the start of the labor? And so you can see here's fetal growth. It distorts the myometrium, which causes an increase in prostaglandin production, which increases the excitability of the uterine musculature. Okay? And that's going to cause labor contractions to occur. You should be able to answer the other 4 questions. I can promise you that you will see this chart on the next on the next lecture exam. All right. Hope you enjoyed the lecture. If you have any questions, feel free to send me an email or check in on the discussion board on Blackboard and get those answered.